Lots of microphones. Anyways, thanks very, very much for inviting me here, Martin and Larry. It's really a great pleasure to be here. And I was fortunate to get a few great days of skiing at Whistler before I came down here, so much the better. And luckily, we have to go to Seattle after this, so my Northwest swing will be done in one trip. So I'm going to talk about a fairly scientific topic today, and I don't have, it's not too many slides, so hopefully I'll be able to go through it slowly enough so that even those who are not scientists will be able to understand. And I think it's an important topic because we're always reading about stem cells, and in the United States, especially in California, we've got this new $1 billion research program funded by the state. All the voters of the state of California voted for a $1 billion borrowing spree. We borrow a lot of money in the States to pay for research in stem cells. Largely because our federal government, as you I'm sure know, has outlawed the study of embryonic stem cells to a large degree. So, and one of the major foci of the stem cell research that's going on at UCLA is actually cancer stem cells. So I'm going to talk to you about cancer stem cells, and then I'm going to talk to you, show you why we believe that they're very important for targeting cancer, and why potentially we've actually been approaching cancer perhaps through the wrong approach by not focusing on the right targets. And then finally, I'm going to spend most of the time talking about PSCA, or prostate stem cell antigen, which is a protein that's present on the surface of prostate. I'll call them stem and progenitor cells. I'll show you how those two things are different. And I'm going to talk about the translation of this discovery, which, as Martin said, we made in my lab about 8 years ago, and how that's actually already now come into the clinic. And there's a phase 1 trial of an antibody directed against this protein that's actually in the clinic on the East Coast at Memorial Sloan Kettering and Johns Hopkins, and which is being co-developed by Merck. So I'm going to show you basically how we've taken this whole concept of stem cells from the lab all the way into the clinic, and I think it's potentially at least a way or a paradigm for thinking about how to treat cancer. So what are stem cells? So basically stem cells, as I'm sure you do know, are the cells from which all other cells come. So there are embryonic stem cells from which we all develop, but then each organ has its own stem cells from which it develops. And so the hypothesis about cancer stem cells is that cancer also arises from a single cell that's transformed, which we call a cancer stem cell. And that this stem cell is actually what maintains cancer. And I think a good way of thinking about this is androgen-independent or hormone-refractory prostate cancer. One of the ways to think about it is imagine that you've got prostate cancer, you castrate a man with this disease, and virtually all of the cancer cells will die, electrical apoptosis, and die. And what survives is believed to be the cancer stem cell population, which then has hormone-refractory capacity and then can grow. So some of the features of cancer stem cells are that they're able to divide and renew themselves, and then give rise to differentiated progeny. And those would be the mature cells of the prostate that secrete PSA, et cetera, et cetera. Cancer stem cells, the idea is that if you took a single cancer stem cell, it would be able to form an entire tumor. And indeed, in breast cancer and glioblastoma, it has been clearly shown that you can identify a cancer stem cell, take that single stem cell, put it under the skin of a mouse, and get an entire tumor with all of the features of a tumor. And the idea ultimately is that instead of targeting the progeny or the differentiated cells in a tumor, what we need to be doing is trying to isolate this cancer stem cell, learn about it, and target it, because if we can target it, we're targeting the cell that actually is important or crucial or critical for the maintenance of a tumor. And so that's the idea of cancer stem cells. And if you have any questions during the talk, please feel free to interrupt me, since at least for some of you this may be kind of a novel concept. But I'm going to go through and I'm going to tell you what we know about stem cells in the prostate. Now, most of the data about stem cells in the prostate is known not in humans, unfortunately, although we're starting to learn about cancer stem cells and stem cells in humans, but from the mice. And this is a complicated slide here, but I guess I don't really have a pointer, so except for here. but I'm going to walk you through little parts of it. But basically, this is work that comes from actually UCLA and also from NYU. There are two investigators who simultaneously published on the identification of prostate stem cells in the normal mouse prostate. And what they found is that you could identify this population by the presence of a protein that's present on the surface of those cells called SCA-I, which stands for stem cell antigen 1. And this is a protein that actually seems to be a feature or marker of stem cells in many different organs. And so it was kind of an almost obvious choice for looking for stem cells in the prostate. And some of the key features of this population are present on this slide. Number one, the localization of these cells in the prostate. It's found that most of the cells localize to this area of the proximal prostate duct. This is the mouse prostate here, and it goes from the proximal to the distal area. And it's found that most of these stem cells marked by SCA-I are located in the proximal area. I'm not going to go through all of this, but I'm going to show you here, I think, the most key feature. The hypothesis that I just started at the beginning telling you is that if you were to castrate a mouse or castrate a human, that after the majority of the differentiated cells would die, basically all the cells that require androgen for their survival, that secrete PSA, all the differentiated progeny, would go ahead and die, and that you would enrich the prostate for stem cells after castration. And indeed, what Owen's group was able to show, if I can get this pointer to work, is that after castration, the percentage of cells that express, that have this SCA-I protein, increases dramatically from 13% here on the left to 66% on the right, again suggesting that this population has features of a stem cell population. The other hypothesis about a stem cell is that you could take as few as a single cell, or certainly a small number of cells, and that you could recapitulate, basically form a new prostate by just using a single stem cell. And so what Owen's group did is they separated all of the cells in the prostate into the cells that had SCA-I on them and the cells that did not have SCA-I. And they were able to show that by taking these cells and putting them underneath the capsule of the kidney, if you took the SCA-I positive population, those that have SCA-I, you could basically form a new prostate, shown here. But if you took the population of cells that did not have SCA-I, you basically did not get a prostate again. So again, showing that this population of cells has the features that are characteristic of stem cells. They have the ability to basically renew themselves and basically give rise to an entire organ. Now, what happens? So initially you start off with a stem cell. The stem cell then gives rise to progeny, which are called transit amplifying cells. So these are cells that are not completely differentiated. They don't necessarily express PSA, but they are still replicating. And so basically you have a kind of a ladder, a hierarchy of cells that start from a stem cell, and then you have these cells called intermediate cells, and then you finally have differentiated cells. And this will become obvious why I'm talking about this in a few minutes. But this is a paper that my group published back in 2002, showing that you can actually use what's called GFP, which is basically a green protein. It's what makes fireflies light up. And you could actually use this as a way of marking cells in the prostate. And you could show that during different periods of time, different cells light up. And so using PSCA, which I'm going to talk about more, as our marker, which basically which is marked here by the green. In the normal mouse prostate, you can see the green here, these very distal tips of the prostate. After you castrate a mouse, and essentially you just have stem cells, you see no green. And then you give testosterone, which is what leads to rapid growth of the prostate, causes the stem cells actually to start growing. And here now you see a very wide or scattered population of green cells. So this is a marker of this very proliferative population. It's not a stem cell, but it's what we call an intermediate cell. And it's actually marked by this green protein here. And so this is just another marker of a different population that I wanted to show. It's a good question. The reason we call it PSCA, prostate stem cell antigen, is because structurally it's very similar to the protein I was talking about, SCA-I. And so before we knew whether it actually was a marker of a stem cell or what it was actually a marker of, we called it prostate stem cell antigen simply because it's so similar to stem cell antigen, and we found it mostly in the prostate, or actually only in the prostate. And that's why we called it prostate stem cell antigen. So what we think we have now in the mouse, and actually partially in the human, is we have a series of different markers present on the surface of the cell that can be used to identify what appears to be a population of stem cells, and then a population of what we call transit cells or intermediate cells, and those are basically the cells that are the progeny of stem cells, and then ultimately these give rise to what are called luminal cells, which are the differentiated, mature cells of the prostate. And these different cell populations are marked by different markers. So SCA-I seems to mark a stem cell population. PSCA, which is a surface protein, again, which we discovered, seems to mark this amplifying population. And then PSA, which we're, of course, most familiar with, seems to mark the luminal or differentiated cells. And the question becomes, where does cancer come from? Does cancer come from this cell? Is this cell transformed by an oncogene or loss of a tumor suppressor gene? Or is it this cell? Or is it this cell? Well, classically, it was believed because prostate cancers express PSA and they have the androgen receptor, that cancers arose from here. But as I've shown you a little bit, and I'm going to show you in the next few slides, there's more and more data that suggests that cancer comes from one of these two populations of cells. And this is what it turns out to be in breast cancer, glioblastoma, and others. And what it turns out is it may be that some cancers arise from transformation of a very early stem population, and others may arise from transformation of a later intermediate cell. And we're just starting to learn about this in prostate cancer. We really don't have the answer yet, but the goal is really just to give you an idea of kind of what the field is thinking right now. So the idea, as I just said, is that cancer can derive from one of these very early populations, and then it can differentiate, giving rise to tumors that have all the heterogeneity that we think of when we think of cancer. And that if you then treat a cancer, like with chemotherapy, what happens is that you kill the vast majority of these cells that form the heterogeneous the cancer mass, but you may not be able to kill the stem cell. So ultimately, the idea is that for us to be really successful in treating cancer, we can't just treat the 95% of cancer cells that we kill, like with castration, and then come back again. We've got to really think about identifying what it is about the cancer stem cell that enables it to survive all of the insults that we throw at it with our current therapies and target that population. And what is the evidence? So here's some evidence that stem cells and these antigens, these proteins that we find in stem cells, are actually present in cancer. So this is an experiment done by Hong Wu, who's really a brilliant scientist at UCLA, together with Owen's group. And what they did is they have a mouse model that develops prostate cancer. And this model occurs, or this cancer occurs because of the loss of a tumor suppressor gene, which I'm not going to get into, called PTEN. And if you look at these tumors, and you look at the population of cells that express this protein, SCA-I, in a normal prostate, it's about 16%, but in the cancer, it's about 89%. So there's an expansion of this pool of cells that expresses these features of a stem cell, or at least some of these features of a stem cell. And the same is true of PSCA, which is why we're so interested in it. So here's a model that we did in our lab where we took that green mouse I showed you, and we bred this green mouse to another prostate cancer-forming mouse called the TRAMP mouse. And the important point is that we found that in the normal mouse, there were very few green cells, but in the cancer, the number of green cells expanded tremendously, which again shows that this marker of now an intermediate population of stem cells or progenitor cells is expanded in cancer, giving us, leading to the hypothesis that maybe you can target some of these proteins. Maybe one way to get at the stem cell population is to target these proteins, which now in cancer are probably present not only on the cancer stem cell, but many of the more differentiated progeny of that tumor. And this is just another model showing, again, in the PTEN mouse that I just talked about briefly, which is not important how it's formed, but basically, again, the percentage of cells that express this marker PSCA in the control mouse all these little dots to the right are the cells that have this protein on it. And in the normal mouse, there aren't many, but in the tumorigenic or in the tumor, there are many, many cells that now express this marker. So that's the mouse. And so I'm going to now shift from the mouse to the human and start telling you about how we've gone about targeting this one protein, prostate stem cell antigen, but certainly there are likely to be other proteins on stem cells that we can target with antibodies or with small molecules or whatever it happens to be. And again, the idea is that in a tumor is formed of a complex mass of cells, including stem cell population and differentiated cells, and that to really kill cancer, we've got to really focus more on these red cells, which are the cancer stem cells, and not completely on all these other white cells, which may not be crucial to the maintenance of a tumor. We've got to figure out what maintains a tumor, what enables a tumor to survive chemotherapy, castration, et cetera. And work here at UBC obviously is focused on this as well by trying to understand what causes hormone-refractory prostate cancer, because hormone-refractory prostate cancer, at least in my mind and many others, may result from the selection of this cancer stem cell. So now getting on to PSCA. So PSCA, initially we found actually in humans, and then we went and found it in mice, and then we started working on this all this stem cell hypothesis. But since we cloned it many years ago, we've really thought about how can we take this protein that's present on the surface of a prostate cancer cell and get from point A to point Z, which is actually getting something into the clinic. And so I'm going to really go through, in the rest of this talk, I'm going to go through the story of how we started by identifying this protein, how we looked at the protein in prostate cancer, how we've tried to understand what the target does, how we made an antibody that can be used as a drug against this target. This is good. I've got a virus and I'm starting to lose my voice. And then I'm going to take you all the way through the story to the identification, to the clinical trials of this antibody. So PSCA, this is the protein here, and it's just to show you that this is a protein that's present on the surface of cells, because it has a sequence here which basically shows that it's attached to the membrane. This protein is expressed in normal prostate and in prostate cancer, and so this is immunohistochemistry of a series of prostate cancers that we looked at, showing the expression of this protein in virtually all prostate cancers. So, and this is basically the brown staining here is indicative of the expression of this protein in prostate cancer, and it's present in Gleason 4 tumors, as in number A here. Gleason 3 tumors at lower levels, and again Gleason 4 tumors here. And what we found is that the higher expression of this protein, it correlated with its adverse outcome. So tumors that were Gleason 4 or 5 tended to express more of this protein than tumors that were Gleason 3. And that kind of goes with this idea that PSCA may be marking a cancer stem cell or a progenitor cell, since the idea is that you think that this population may be enriched or present more highly in very aggressive kinds of prostate cancer. So this was basically a study looking at about 180 prostate cancers that we operated on at UCLA, and for which we had clinical data showing that the higher expression of PSCA, the more likely the cancer was to recur after surgery. Of course, it's not enough just to look at the localized tumor. Ultimately, what we want to treat are metastatic tumors. So we published last year a paper looking at the presence of this protein in metastatic cancer. And luckily, we were able to get tissues from University of Washington that has this very large series of autopsies where they basically took patients who died of prostate cancer, collected their tissues, collected the tumors from around the body, including the bone and lymph nodes, and we were able to get some of these and look at them. And as you can see here, in these bone metastases on the right and this liver metastasis on the left, very, very high presence of PSCA in these tumors. But we didn't see it in every metastatic tumor. Here's a lymph node where we actually saw very little or no expression. And we tabulated all of this here I hope I don't have it here. We tabulated this by showing ultimately that about 80% of prostate cancer metastases have very, very strong levels. What we wanted to do here, and what this slide shows, is we wanted to prove not only by staining, looking at the protein, but we wanted to look at the RNA from which protein is derived in these metastases to be sure that the protein, that PSCA is really there, and we wanted to quantify how much is there. And basically what this shows is looking by a PCR technique, basically quantifying the amount of PSCA in tumors. We can find very, very, very high levels of PSCA in many of these prostate cancer. These are all different prostate cancer metastases. We see very, very high levels. And the important point here as I go through our data is that, thanks so much, is that the experiments I'm going to show you in mice, we used a human prostate cancer xenograft or cell line called LAPC9, which is here on the very far left. And we wanted to quantify the amount of PSCA in tumors to see if it was similar to what we see in the model system that we've used to look at our therapy, our antibody therapy. And so what you can see is that in more than half of the tumors, the level of this protein is actually significantly 3 or more fold than even in this model system that we use. And so I'll come back to that point as we go on. So what we did at this point is and what we've used for all the experiments I just showed is we made an antibody that can recognize this protein. And the reason we made an antibody initially was just to study the protein, because to study a protein you need something that recognizes it. But my long-term goal when I actually started in the lab was to identify proteins that are on the surface of cells because they're amenable, they're exposed on the surface, so they can be targeted with antibodies. And as I'm sure you know, antibody therapy for cancer is a very hot area. Herceptin, Avastin, there are now many antibody therapeutics for cancer. So the idea we had is if we can identify cell surface proteins, we can make antibodies against them, and potentially those antibodies could actually be drugs. And they're actually in many ways the easiest drugs to develop because basically all you have to do is put the protein in a mouse, the mouse makes an antibody against it. So they're generally, conceptually very easy. So what we did is we made a series of antibodies, and we went ahead and tested all of them. And in this case, we're testing one called 1G8. And 1G8 is actually the antibody that seems to be the most promising among the antibodies that we made. And what we did is we took these tumors called LAPC9, which is basically a human cell line that we developed at UCLA that comes from a patient, actually of mine, who died from prostate cancer. So it came from a patient with hormone-refractory prostate cancer. And what we did is we implanted this cell line in mice, and then we either gave the mice just saline, PBS, or we gave it the antibody. And this was done by implanting, actually in this case, we actually implanted the tumor into the prostate because we wanted to look not just at, we wanted to look at it in a quote-unquote natural setting, since of course if we're going to treat humans, we're going to treat humans with tumors in their prostate. So we implanted the tumor in the prostate, and what we did is we measured PSA. These tumors make PSA, so we could actually follow the progression of the tumor by looking at PSA. And you can see by giving antibody, you basically inhibit the growth of this tumor, as shown here. And by giving antibody, the median survival of the animals increased by about 50% as well. So in this experiment, this was a surprise to us. We actually thought that we would have to attach a radioactive isotope or some kind of drug that would kill cells, but it turns out just by blocking or binding to this protein, the antibody is able to inhibit the growth of prostate cancer. And not only did it inhibit the growth of prostate cancer in the primary tumor, but it completely blocked metastasis of these tumors in mice. And so that's what's shown here. This LAPC9 tumor, if you put it orthotopically, if you put it into the prostate of mice, it normally spreads to the lung. And you can take the lung, section it, and then stain it for PSA and see these metastases. And if you do that basically in the animals that were given the antibody, we saw a few tumors only in about metastatic tumors in about 1 out of every 9 mice, 12% of mice. And in that mouse, the number of tumors was far, far fewer than in the controls. So it almost completely blocked the spread of this tumor in mice. So we have evidence that by using an antibody directed against an antigen that we believe is expressed in most prostate cancers and most prostate cancer metastases, and may be a marker of an earlier progenitor population, this cancer stem cell, that we could actually have an effect on the growth of cancer. So we wanted to, the next thing that our lab focused on was not just making the antibody and saying yes it works, but we really wanted to understand a little bit about why it works. Because our hypothesis is that if we knew how it worked, we might be able to figure out combination strategies, what to combine this with, that would actually be better than just using the antibody alone. And also because we wanted to know whether it was working via the immune system or via some other mechanism. So antibodies, as you probably remember from medical school or just hearing about, basically have, they work in a couple of ways. So they've got two, it's shaped like a triangle. So they've got two recognition sites here, or antibody recognition sites, which actually bind to the protein itself, shown in red here. But they also have a tail, or the FC region, which is able to bind to receptors on the immune system and thereby get the immune system to kill the target cells. So that's how our humoral immune system works. And so there have been a number of studies, such as this diagram here, which comes from a paper by a guy named Jeff Ravitch, who's at the Rockefeller University in New York City. And he actually did some work with the antibody Herceptin and rituximab, which is used to treat lymphoma. And he did studies to see whether or not these antibodies are killing their target, the cancer, by binding to the target on the cell, which is how people hypothesize they work, or actually by engaging the immune system and basically working as a passive immune mechanism. And what he showed is actually, although Genentech and some of the companies that make these antibodies have argued that these new antibodies work completely and only by changing the signal transduction system in a cancer cell, basically by binding to proteins on the surface of the cell and sending signals to the nucleus which kill the cell, it turns out actually that a large part of their activity may also come from engaging the immune system. So we wanted to ask whether or not the antibody that we made, 1G8, works by engaging the immune system or if it works actually by binding the PSCA protein on the surface of cell. And if so, we wanted to then start to figure out what this protein does and how it works and how it's involved in cancer. And we don't completely know the answer to that yet, but I'll show you some of the data that we have and that we just published. So an easy way, if you want to figure out whether an antibody is working via the immune system or if it's working by targeting a cancer cell, what you can do is basically take this antibody and cut it up. And you could cut it up by cutting off the FC part, so it lacks it, so it cannot engage the immune system, or you can basically cut most of it and see whether or not it needs if it needs two recognition sites or a single recognition site. So we did all these different experiments to basically determine how it works. So the first thing we did is we first went to tissue culture, since it's so easy to do things in tissue culture. And we add the antibody to tissue culture because, of course, in the tissue culture, all you have are the cancer cells. There's no immune system. So we asked if the antibody has any activity in culture. And sure enough, in culture, what we see is the antibody can block the growth of the cells. And here it shows that this is just counting the number of dead cells after you add antibody to a plate, and that the antibody basically kills cells. So if you add antibody, if you add nothing to the cancer cell, nothing happens. If you add antibody, about 30-40% of the cancer cells die. So that suggests that it may not be working completely through an immune mechanism, but actually may be binding to this target PSCA on the surface of cells and somehow killing them. And this basically shows here in this slide the same thing, that it basically can kill cells. So this is just showing the percentage of cells that are alive after treatment with our antibody. So there's no antibody here. This many cells are alive. You give antibody, and basically all these, this many cells die, and this is what's left. And what we showed is that you need two recognition sites. If you just have one recognition site, it's not enough to kill the cell. You need two recognition sites, and this is actually common to many of the antibodies that are now therapeutic, such as Rituxan, that the antibody needs to cross-link the protein on the surface of cells. So it needs to bind it in two parts and bring it together, and somehow that's what transduces a message or signals a message to kill the cell. And the next thing we did is we chopped up the antibody to form these Fab2 fragments. So they have 2 recognition sites, but they cannot engage the immune system. So this is the experiment we did to see if the immune system is involved. And this slide here just shows that this, if you cut it up, it still works. It still recognizes, it still sees and binds to this protein on the cell surface. And so this is what's called a FACS analysis. And basically a shift to the right shows that the antibody is actually seeing the protein on the surface of the cell. And so that just showed us, okay, that we can cut this up and make it work. And then we went to the mouse, and instead of putting it orthotopically into the prostate, we just grew the tumors underneath the skin of the mice. And what we can show is that with or without the part of the protein that interacts with the immune system, we're able to block the growth of cancer in mice. So this suggests that the antibody, it doesn't require the immune system to be active. By simply binding with 2 recognition sites to the protein on the surface of the cell, it's able to block it and kill it. And so that's what this shows here. This is the control here of the tumor growing, and then here is in pink is the normal antibody, and here in yellow is the cut up antibody that lacks the FC portion, the part that engages the immune system, and it's able to block the growth. And this is showing that if you take a larger tumor, put it in the mice, that even a large tumor that you put under the skin of the mice, you can essentially block the growth of the tumor by about 50-60% using either the partial antibody or the whole antibody. So this starts to tell us about how the antibody is actually working. And then this is a kind of a cool experiment. This guy, Jeff Ravitch, he made a mouse where he was able to completely ablate the receptor for the FC portion of the antibody. So it's a mouse that lacks the ability, the macrophages and the immune cells cannot actually bind to antibody. So we did the same experiment using our antibody in these mice, and again we're able to show that without a functional immune system, an immune system that can actually bind to our antibody, that the antibody still works. And so this shows us how the antibody is working through a direct signaling or direct binding method. So I'm actually going to go through these slides because I think they get a little bit too scientific, and I'm going to get to kind of where we are now. So I've shown you, I hope I've shown you, that we think that cancer arises from stem or progenitor cells, that PSCA may be one marker of those cells, that it's expressed very highly in almost all prostate cancers, localized and metastatic. It correlates with adverse outcome. An antibody targeted against it can block growth in preclinical models. And so the next step really was to take this obviously into humans, the next clinical step. And so you can do this in many ways. If you just take a mouse antibody, which is what 1G8 is, you really can't use that in humans because our immune system will recognize this as a mouse antibody, generate an immune response against it, and essentially abolish it. So you could give one dose of drug, but after you gave that, it'd be gone. So what people have done over the years is they've gone through a series of steps to make this mouse antibody or transform it from a mouse antibody into a human antibody, making it less antigenic. And you can do that many ways. You can replace the mouse tail, the portion of the mouse antibody that recognizes the immune system, and replace it with a human FC region as shown here. So everything in pink is human, but the recognition site, the thing that actually binds to the protein, PSCA, is still mouse. And that's actually Rituxan. The antibody in clinical use, Rituxan, is a chimeric antibody. And it doesn't generate a big immune response against it, although that's partially because it's directed against T and B cells, so in some ways it's actually downregulating the immune system so that your immune system can't build up a big response to it. But there's the other antibody, Erbitux, which is being used now to treat colon cancer and head and neck cancer, is also a chimeric antibody. So the first step that we did is we wanted to see if we could make a chimeric antibody. The second step we did is we went the next step, which is what Herceptin is, where what you do is you replace almost all of the mouse antibody with human sequences, human protein sequences, and the only thing that you leave behind are the actual small little sequences of the mouse antibody that actually recognize the protein. And this is called a humanized antibody, and shown here. So where just the stripes are human, but everything else is mouse. And then the final kind of antibody, which I want to talk about, is a completely human antibody. And there are now technologies to completely replace all of the mouse elements with human elements. So there's 4 different kinds of antibodies, and actually, believe it or not, we or others have made now all of these against PSCA. So I'm going to show you each piece of data. So the first one we made was a chimeric antibody. And this is actually relatively straightforward, not simple, but straightforward to do in the laboratory. And what we found is that if we make this antibody, it actually, as I'll show you, it works very nicely. The problem is we couldn't make a whole lot of it. And of course, to put this into patients, ultimately, we've got to be able to make a whole lot of it. But as shown here, this yellow is the chimeric antibody, and it retains, actually even has greater activity for some reason against the human prostate cancer in a mouse model than the other. So it at least shows the strategy that you can make a human-like or partially human antibody and start to get it to work. But the problem, unfortunately, we had, which is common of chimeric antibodies, is it's difficult to produce them in very large amounts. So the next step is we collaborated with somebody at UCLA named Anna Wu and a group of City of Hope. City of Hope, which is also in Los Angeles, has a very large cancer antibody program. And so we worked with them to do the much more complex task of actually taking our mouse 1G8 antibody and humanizing it. And the result of that is an antibody that we're now working with called 2B3. And 2B3 basically is everything on it is human. It's just like Herceptin, except for these small regions here that recognize the PSCA protein. We've been able to make this. We've been able to purify large amounts of it, and we've been able to study it, as I'll show here. So the first thing we did is we took this this humanized antibody, and we wanted to show that it still retains the ability to recognize or bind to this PSCA protein. And that's what's shown here. So this is the control. This is the mouse antibody. And then these are actually 2 different humanized antibodies we made, 2B3 here, which I think is our best one, and 3E10. And they retain the ability to recognize PSCA. In addition, they work. This is actually a bladder cancer model that expresses PSCA, and it expresses very, very low levels of it. And in fact, it expresses such low levels of it that the mouse antibody doesn't really work very well against it, but this humanized antibody actually does work pretty well against it. We think it's because this humanized antibody, not only does it bind to it, but it may be able to engage the immune system. So even though the mouse antibody did not need the immune system to work, it may be that the human FC portion of the antibody is able to actually bring the immune system in it, but we haven't proven that. One of the things we wanted to do is we wanted to prove that the antibody actually goes and targets to a tumor and doesn't target to the rest of the body, because ultimately, to be safe, we really want to focus on targeting the tumor. PSCA normally is expressed almost completely in the prostate. It has very low levels of presence in the superficial bladder and very low levels in the stomach. And if you give the antibody, so we label the antibody with iodine and then can image it, we can see here that it can traffic to a tumor that has PSCA, but it does not go to a tumor, here B, that does not have PSCA, and there's very little residual protein left in the body after about 24 to 48 hours. And that's what these measurements show here. So the humanized antibody is active, and it traffics to tumor. And those are obviously the things that we want it to do. Now, where we are currently in terms of this humanized antibody is that we're still trying to work to improve it, because we find very commonly when you humanize an antibody, even though it still retains the ability to bind to the protein, some of the strength, the affinity for the protein, or the strength with which the antibody binds to the protein, decreases somewhat. So we're working with a group at UCSF to what's called affinity maturity antibody, which basically means to make it bind even more strongly. And we actually have a series of candidates right now which we're screening and making that actually seem to be better than 2B3. Because our goal here is, as I'll talk about, I guess I'll talk about it now, our goal is actually to take this antibody by ourselves into human studies. And our goal is actually to use it for imaging. So we can radio label it with an isotope. That's what City of Hope does very, very well. And just with very, very small amounts of this antibody, we can actually see whether or not we get the antibody to traffic to human prostate cancers. And then that's basically the first step, obviously, in getting biotechnology or pharmaceutical industry to be interested in this particular antibody. And I'll explain why we think that's important in a few slides at the end of the talk. But so this is academically, just in our own laboratory, what we've been able to do, taking it from the discovery all the way to now having a molecule which we think is almost ready to go into humans. And so that's where we are right now. Now, we also, UCLA, also licensed this discovery a number of years ago to a small biotechnology company called Agensis. And Agensis kind of followed along with the work that we were performing, and gradually, as we showed that it was present in almost all prostate cancers, and also, I didn't tell you, it's also present in pancreatic cancer and bladder cancer, so they started to get more and more interested. So they worked with a company that called Antigens, and this company has a mouse in which they've replaced all of the normal human B-cell components of the genome. So all the chromosomal regions that encode for our ability to make antibodies, and they replaced them with human sequences. So it's a mouse that's partially human, and if you basically take any protein like PSCA and you inject it into these mice, they will form completely human antibodies against that antigen. So this company collaborated with Antigens to make a completely human antibody against PSCA, and they were successful in doing that. And they have an antibody now that they call AGS-PSCA. And they've been able to show, they reported now, that the antibody recognizes PSCA. It has very high affinity. It binds very strongly to PSCA. And they've been able to basically replicate the experiments that I showed you with the mouse 1G8 antibody. They've been able to replicate those with this human antibody and show that it blocks tumor growth, blocks metastasis. And if you combine it with chemotherapy, with Taxotere, actually causes complete regression of tumors, whereas Taxotere alone in these models is not enough to. So it shows some synergy between this antibody and Taxotere. They don't yet know if it recognizes the same region of the protein as 1G8 does, so this may not be the same antibody as 1G8, which is an important point. And they have not yet done studies to show exactly how it works like we have. So we are continuing to develop our antibody because there's a possibility that the antibodies may function differently. And so I think it's important to develop oftentimes multiple drugs against a single target to find out which works best. But the good news is that this antibody has made it through most of the FDA hurdles in terms of getting it into humans. It's been shown to be safe if given to mammals, and they can produce plenty of it. And it's actually now in phase 1 trials at Memorial Sloan Kettering and Johns Hopkins. All I know about it thus far is it seems to be safe. And this is a phase 1 trial, basically, to look at safety of various doses of the antibody. And so hopefully we'll hear more data about this within the next year or so in terms of whether it's having any activity at all and whether it's safe. Most likely, if it's safe, they'll probably go on to a phase 2 trial combining this with Taxotere. So that's where I'm going to end. I think on time, a little bit early. And these are just acknowledgments. This work, I basically just showed you in 40 minutes about 8 years worth of work. But basically, so there have been many, many people from my lab involved and others. So for my lab, basically a large amount of this work's been done by Zenon Gu, who's been in my lab now and Joyce Yamashiro since the beginning. Zenon made the antibodies. He's done, together with Joyce and Evelyn, have done much of the animal work and have quite a few publications on this work. Anna Wu, who helps us with all of the humanized antibodies, she's a brilliant biologist. Owen Whitty, with who I actually started out, who is my lab mentor. Sherry Morrison is an antibody expert at UCLA who helps us make chimeric antibodies. Many pathologists